me at all. Okay, so this week you guys are going to do the easier of the two muscle labs, which is good because the other one was a lot. Okay, so what you're going to do in lab this week is axial musculature, right? There are fewer muscles. There are fewer attachment points that you have to learn for the muscles, right? And they are easier to identify. So what you will do is after you've done the axial muscles, you're going to go back and review the appendicular muscles. But again, I'm going to reinforce this for you as you're all feeling like your confidence get a little shaken when you go through those. It happens to the best of us, man, I promise you. You need to scaffold your learning in, because last time what you did is you got confident with a lot of the muscle names. Maybe not these little bastards, but the rest of them, they're totally cool. You're like, I know where the damn Terry's muscles are. Shut up. You do. Yeah. Minor over major. Yes, minor over major. So what you need to do this time is you need to, when you look at the muscles, you need to force yourself to scaffold in some more information, which is actions, right? What I would probably do is either do attachment points and then actions, or maybe actions and then attachment points, whichever you want to do. And so like you go over a table, the first time you go over it, you just review the muscles. But the second time you review the muscles with the actions. The third time you do review the muscles with the actions and the attachment points, right? And you do all of that on the cadaver because the more comfortable you are with the cadaver, the better off you are. What was your question, comment?
So you think, or do you know that the cadavers are the best way? Like pictures don't help with muscles?
No, the pictures do help with muscles, but if you're in lab, definitely use that because those, they totally help. I will caution you against passive studying for difficult material. Do you know what I mean about passive studying? Just reading and flipping through things gives you a false sense of confidence. You need to do something active. You either need to be writing it, you need to be labeling it, you need to be coloring it, you need to be doing something active. Looking at the tables that are all filled out is not that good for you in later stages of studying, but definitely pictures are much better than nothing, right? It's just that the cadavers are harder than the pictures, right? So when you're in front of them, you need to use them, right? I also, and I know some people don't, I like to look at the models in the lab before I look at the cadavers so I can get this idealized version. I even label all the muscles on the freaking forearm, right? And then I take that model over to the cadaver and I'm like, this is what it should look like. What the hell is going on there? No, it kind of looks like it, right? So whatever you can do to give yourself some confidence. 2D is a little hard for me. I usually like 3D or models or whatever, but go ahead.
It's hard because everything's the same color.
I know.
In a cadaver, there's no color coordination.
That's true. There is no color coordination. I'm sorry about that for you visual learners. There isn't. It's all meat colored. That's what it is. Yeah? Yeah? Okay. So, and also, some of you guys joined the anatomy study group online. They post lots of cool quizzes and stuff like that there. And don't be afraid to rummage through all the stuff on SLCC anatomy because there's lots of helpful stuff on there. But there is no substitute for time and active learning, right? You have to get actively engaged in the process. And it's a lot. It is. But you're a bunch of little badasses and you can totally do it. We learn more muscles than the U does. And we also learn, they don't learn any of the nervous structures. They don't do any brain structures. So you guys are going to leave here and they're going to say they're better. And then you ask them some muscles that they don't know and we will know who's better, okay? Okay, so let's talk about these just briefly. So these are a bunch of the muscles of facial expression. You don't learn them all, right? You don't learn all of the muscles of anything, right? And you learn enough. Can we agree that you learn enough muscles? Yeah, it's enough. I'm pretty okay with it. So, but I do want to get you guys used to what some of the muscles do as you start to read the list today. So, so let's talk about, they call this one the occipitofrontalis, but they separated those two muscles, and this is the actual frontalis, and this is the occipitalis. So if you use just the frontalis muscle here, you do this. Yeah? Yeah, everybody do it? If you've had Botox recently, you can't do that, okay? And I haven't recently. It's about time again. So that's the frontalis muscle right there. Its little partner is right back here called the occipitalis. How about the orbicularis oculi, right? It's closing your eyes. The muscles of facial expression do not, for the most part, insert onto bones at all. They just insert onto the connective tissue of your face. They end up being really evolutionarily important because as we got more bald, we actually gain the ability to communicate non-verbally with one another way, way better than other organisms. So you can completely change the meaning of a word with just your facial expressions, because she can say something to me and I can say, yeah, right. And it means something totally different than, yeah, right? Even if I held the inflection of my voice totally a constant, you can completely change the nature of the meaning with facial expressions. Interestingly, people with autism, and my family has tons of autism on my husband's side of the family, they do not pick up nonverbal facial cues as readily. You can totally train a person with autism to pick them up, but it's more analytical than intuitive. So you're going to have to tell them that when somebody makes this face, for instance, my brother-in-law, Rick, when somebody makes this face, Rick, they're probably confused with what you're talking about. Orbicularis oculi, the zygomaticus major and minor. There's two muscles right here. You learned this week. It's still minor over major. Major goes more medial. Minor goes more lateral. How much control do you guys have with those? What can you do? Right? Can you do both sides? If you're just doing the, yeah. Oh, she can do both sides. I can't do both sides. I can only do one side. And if you're just using the minor, you can get that little thing going there in the middle, right? Middle. Do it. Move your damn faces. You got a study tool on your face. Do it. Right? So that's the major and the minor. The buccinator and the orbicularis oris are being used over here. So the orbicularis oris closes the lips, right? The buccinator is this one that goes here. If you use the buccinator and you also put air in your mouth, then you'll do this. If you use it and you don't put air in your mouth, you can suck your cheeks in. So the major and minor. The orbicularis oris, ideally when you're eating, you will use both this one and the masseter, because you will close your jaw and your lips, yes? And then people will go out to eat with you again. I've got a 20-year-old who still insists that that's not necessary, which is why he's never going to have a girlfriend. So, orbicularis oris, and then the platysma. Everybody do this. I feel it. That's our platysma. Right there. Okay. So those are some of the muscles of facial expression, not all of them. There's some pretty good ones in there too. See, this is the one that I told you guys that depressor anguli oris. What's the sound effect? You know the sound effect. I'm so proud. Okay. So we are now going to change gears a little bit, and we're going to move into muscle structure, deeper structure, and then into the stuff that leads into muscle physiology. But before we do, let's just give you guys a little bit of intro to how muscle tissues are different than other tissues. So the primary thing that makes muscle tissue different than other tissue is that when stimulated, all three types of muscle have a characteristic that no other tissue does, which is called contractility, meaning that when you stimulate muscle, what they will do is actually shorten, and no other tissue does that. What's happening when muscle shortens, by the way, is that I'm taking chemical energy in the form of ATP and converting it to mechanical or kinetic energy in the form of movement. Yeah, that's what I'm doing. And you guys have learned enough in your science classes before to know that energy conversion tends to be really inefficient. And what is released as a byproduct because of the inefficiency of energy conversion? Heat. Yes, this is part of the reason that muscle contraction is a really, really dependable way to heat up your body. And you can either do it incidentally, for instance, when you work out, your body heats up, or you can do it purposefully, like, for instance, when you shiver to cause heat production, right? Okay, so muscle also has some other characteristics. It has what's called excitability, and excitability means that it can respond to an electrical stimulus. Muscle and nervous tissues are electrically excitable. And the reason that is because they have these little electric channels on their surfaces. So if you, for instance, electrocute somebody, which tissues are going to get fried? Muscle and nervous tissue, right? That's them. But muscle has two other characteristics. It's also got what we call extensibility, meaning that you can stretch it. So you can stretch your muscles because there's connective tissue within them. We'll learn about the connective tissue today. but it also has snapback because there's a little bit of elasticity within the tissue, meaning that you can't, for instance, if you go to a yoga class and you really stretch your hip flexors, they're not going to become flaccid and not snap back, right? Okay. And then I do want to remind you that you learned about muscle tissues and the differences and similarities between them back in the histology chapter, which ones are striated, which ones are uninucleated, what the shape is. So go back and look at that before you take your next test, which is no time soon, so don't worry. Okay, so a little bit about the functionality of muscle tissue. Of course, what everybody attributes to muscle is motion, yes? But that's definitely not all that it does. So yes, all three types of muscle tissue move or move things around. But muscle is also important for postural stability. So if you, for instance, if you were quadriplegic, could you hold yourself upright in your chair without assistance, right? No, you couldn't, right? You would actually need assistance to hold up. So postural stability is muscular contraction just as much as motion is. And then muscle also stabilizes your joints. So if you have a joint that has been damaged, what you need to do is strengthen the muscle around that to stabilize it. Muscle is used for heat production, shivering, right? Involuntary response to heat up your body. Muscle is also in some places supportive. So the muscles, your abdominal muscles, yes, they do movement, but they also squish your guts back against your body wall, right? And they're supporting your guts in there. So if, for instance, you have an emergency abdominal surgery and you cut straight through them, you're going to lose some of the structural integrity. You're going to have a pooch even if you don't have subcutaneous fat to speak of, right? And the muscles here, these are supportive. They're keeping your junk up there, right? So those muscles, the pubococcygeal muscles, are keeping your junk up there. If you have a whole bunch of big-headed babies, you can rip the living hell out of those, and you can have what's called uterine prolapse, in which your uterus flops out. Don't you think they should teach that in middle school if they don't want them to have sex?
Yeah.
Because women will be like, I want my junk up there where it goes, right? You keep that thing over there. I'm going to tell my son about that. It'll gross him out enough. It'll put off sex 2 more years. Guarding openings, sphincters, skeletal smooth muscle sphincters, control openings, and then keeping your fluids moving. Muscle, obviously cardiac muscle does, duh. But then the smooth muscle in the walls of the tubes, right, all through your body keeps fluids moving. But then, although it's counterintuitive, skeletal muscle also keeps blood and lymph moving because so let's imagine the veins that are going from my feet all the way back up to my heart, carrying blood back to the heart. They've got a few things working against them. First of all, they're really far away from the pump, so they're going to be super low pressure. Second, they're running against gravity, right? Okay. So what helps blood to return through veins is that skeletal muscle can squish on the vessels, and those long veins that are going back towards your heart have one-way valves in them. So if you squish on it, the blood can't go backwards, so it actually has to go forward. So the combination of skeletal muscle contraction around them and the one-way valves will actually help that. Does that make sense to you? For that reason, when you are physically active, you not only have better blood flow because of what your heart's doing, but also better blood flow because of what your skeletal muscles are doing. Does that make sense? Okay. All right, so now let's get into the organization of a skeletal muscle. Okay, so with a whole skeletal muscle, first let's just do a little terminology. When you see the prefix MYO or the suffix MYS, it refers to muscle. Okay, so you will see those quite often throughout this set of notes and then also through physiology. And you guys remember, don't you, that you can use the term muscle fiber and muscle cell interchangeably, right? Okay, it's not the same thing as saying a collagen fiber, which is a protein, right? A whole muscle cell is the same as a muscle fiber because they're long and skinny. Does that make sense? Okay, and then in this set of notes and in phys, you will also use this prefix sarco. Sarco means flesh, and it's used to refer to microscopic muscle structures and submicroscopic. So typically, and there are exceptions to this, when you're looking at big muscle structures, they have myo. And when you're looking at teeny tiny submicroscopic muscle structures, they have sarco, okay? Okay, so first thing that we are going to talk about is the fact that a whole muscle is not just made of muscle tissue. A whole muscle is actually an organ, and it's got other tissues associated with it. This is a whole muscle. I can't figure out which head of the biceps that's supposed to be, but one of the heads of the biceps maybe, right? Because that looks like the humerus, but I don't know which one it's supposed to be. Okay, so this whole muscle that you're actually looking at right here is, of course, skeletal muscle tissue, but there's also connective tissue associated with it because the muscle is going to be under tremendous stress when, for instance, you have somebody who's trying to lift up something that's 400 pounds. Whether they lift it up or not, it's going to put tremendous stress on the muscle to do so, right? So how do you hold the structural integrity of a muscle when you're putting it under so much stress? Well, you have layer after layer of connective tissue wrapping around it. So let's start with the outermost connective tissue. The outermost connective tissue wrapping around the muscle is called muscle, exactly. You're going to come back on, or you're not going to? Do it. Okay, there we go. Implying muscle. Everybody good? So epimysium is wrapping all the way around the muscle. And what kind of tissue would you make that if you got to design a muscle? What would you make it? Say it loud, Bridget. Yeah, that's what I would make it of too. It is dense, irregular connective tissue wrapping all the way around the outside of the muscle. How do we make sure that the muscle is really strong also in its attachment to the tendon and its attachment to the bone? Well, what we do is instead of making a seam, right? So where do your clothes tear? On the seams. So let's make it a continuous fabric instead. So the dense irregular connective tissue around the outside, what I'm going to do is I'm going to weave it into the tendon, which is made of what kind of tissue? Dense regular, yes? Okay, so all I want to do is run those collagen bundle fibers together so that there's not actually any seam between the dense regular and the dense irregular right here. And then it's very unlikely to pull it off, right? And then I'm going to take the tendon and I'm going to weave it into the outside of the bone. Do you remember what tissue wraps around the outside of bones? Dense irregular again, right? So what I'm going to do to make that association really strong is to weave it in. Again, those bundles of collagen fibers, so that really there's a period in which the tissue just changes from one to the other without, even interrupting the orientation of the fibers, just redirecting the orientation of the fibers. So for that reason, you don't often see a muscle torn completely off a bone, even when it's tremendously stressed. I'm not saying never, because every damn thing that could happen has happened, right? But you get a lot of contraction strength with very few tendon tears, comparatively speaking. So it's a really strong association that you have there. Okay, so outside is epimysium, but then make it even stronger by folding this outer tissue called epimysium, thin it out a little bit, and fold it in and wrap around every single bundle of muscle fibers. Do you remember I told you guys that you can't see an individual muscle fiber with the naked eye, but you can see a bundle? That's why you can tell that the rectus abdominis is rectus, right? So a bundle of muscle fibers is called anybody remember? Fascicle. Yeah? Fascicle. So now what I'm going to do is I'm going to take this epimysium, thin it out, and wrap it around every single fascicle, and I'm going to call it perimysium. Okay? And really what it is kind of a much thinner, dense, irregular, almost like an areolar, but with more collagen fibers in it. Okay? It's not a tissue that you learned specifically. So I'm going to wrap this around as perimysium. Good? Okay? It's going to wrap around every single fascicle. But then I'm still not done because I want it to be stronger still. And now what I'm going to do is I'm going to fold in the perimysium, thin it out a little further, and wrap it around every single muscle fiber, right, as endomysium, right? So I have real continuous connective tissue outside, then the wrap around every single fascicle, and then wrap around every single cell within every single fascicle. And so the muscle association, the structural integrity of the muscle is really, really strong. Everybody okay? So what order does it go in? Epi, peri, endo, right? Upon the muscle, around the fascicle, right? And then endo, which is inside, right? Epi, peri, endo. Good? And by the way, they could have really done peri and epi either way if you ask me, but I just want you to remember what order it is, epi, peri, endo. Okay? So the epi, the peri, and the endo are continuous with one another, and then they will be continuous with a tendon, which will be continuous with the periosteum of the bone, right? So you get this really strong association. Okay, so now next thing to talk about is a little bit about how to control muscle tissue and then how vascular muscle tissue needs to be. So in general, what's going to happen, kind of similar to what we said before, is Remember when we said that there's a bone and it's usually going to have an artery and a vein coming in somewhere along the midline, right? And that's called the perforating artery. Well, with a whole muscle, it usually has at least one big artery going in and one big vein coming from it and one big nerve going to it. Now, if it's a big muscle, it may have more than one, but generally speaking, it's going to have at least one association right there. But then, here's what's really important. Every single cell within that muscle has to have really, really good blood supply because this is really energetically expensive tissue. So what's going to happen is you are going to have to sort of migrate that deep within the connective tissue and then split it into arterioles and then split it into smaller and smaller capillaries until every single cell, every single fiber has a capillary net completely surrounding it. So the tissue is highly vascular. That is why your cow muscle that you call a steak, yes, is red because it's incredibly vascular tissue. Does that make sense? Every single cell, and there could be 100,000 of them, right? And you're doing the cross-section of a cow muscle, which is a steak. Every single one of them has capillaries sort of snuggling around it. So it's really vascular tissue. And the tissue is also highly, highly innervated, meaning that, and this is important for you guys to understand, in order for a skeletal muscle fiber to do anything, so let's look at what the fiber looks like. So this is a skeletal muscle fiber right here, okay? Whole muscle fiber. It looks weird. It is a cell. It doesn't look like the cells you covered in 1610. We'll talk about why in just a second. But every single cell within this whole muscle has to be directed what to do every damn time. It has no initiative whatsoever, kind of like a teenager, right? No initiative, right? Not going to do anything unless you were specifically asked to do it, at least my teenagers, right? They will do it when you ask them. So what's going to happen is you have to command each one individually. So if we look right here, this is 3 skeletal muscle fibers. This is the neuron. The cell body is up there somewhere, right? Axon of the neuron that is going to innervate these 3. Okay, so what I'm saying is these 3, if this is damaged, they will never do a thing unless it actually regenerates, okay? But what if this one is damaged? Then this one is paralyzed. Does that make sense to you guys? Okay, now that's different than cardiac muscle. Do you have to tell cardiac muscle every single cell what to do? Why not?
It's involuntary.
But I don't even mean consciously or voluntarily. How do you get around having to talk to each individual cardiac muscle cell? They can talk to one another via what? Gap junctions, yes, right? So if you 3 right there were cardiac muscle cells and I gave her a command to contract, she would both contract and send on the command, right? But with skeletal muscle, every single skeletal muscle fiber has to have individual innervation or else you will have paralysis of that fiber. Just a second. That means that if you've ever had nerve damage or known anyone with nerve damage, yeah, you can get complete paralysis if you damage the entire nerve, but if you didn't damage the entire nerve, you can have muscle weakness because you can't contract every single cell. Go ahead.
So how and why do muscle spasms happen then?
That's a phys question. It's primarily due to ion or hormone imbalances, usually potassium or calcium. But it's a good question. It's just not something I can cover right now. Okay, go ahead, Mazzy.
So is it like kind of like a ripple effect? Like if the middle one is damaged, then the end one is damaged?
No, So these are all working together. They're all going to be controlled simultaneously because they're all controlled by the same neuron. So these are what's called a motor unit, by the way. This one is independent of the others, right? With just because I can actually damage that particular part. So they don't talk to one another at all in skeletal muscle. They are all directly commanded by the nervous system, okay? So that's why any nerve damage with skeletal muscle, you don't know whether you're actually going to re-innervate or re-get the control of the skeletal muscle. And these axons grow really, really slowly. So usually it's around 2 years before you know whether you're going to have any complete reinnervation of a nerve. So a responsible physician after nerve damage would not tell you you're absolutely going to get that sensory or motor function back. They would say, we will know later. You might gain a little bit of it back over time. Usually whatever happens in 2 years is pretty much permanent. Okay, so highly innervated. The site at which it innervates, the nervous system innervates, is called the neuromuscular junction. So this little portion right here where the nerve talks to the muscle is called the neuromuscular junction. In phys, we spend a ton of time on the neuromuscular junction. We talk about what neurotransmitters are released and how they will actually cause an electrical stimulus, and what actually happens there. In anatomy, really all you have to know is the site of communication is the neuromuscular junction. In phys, you actually talk about the functionality. So that's the NMJ, or neuromuscular junction. So each skeletal muscle fiber will only contract when stimulated by the neuromuscular junction, okay? And it's completely snuggled up by capillaries. All right, so now what we are going to do now is we're going to look at an individual muscle fiber, and we're going to sort of start to break that down and see why it looks so funny and what it has to do with the structure and the function. So stick with me for just a second because it's kind of hard to follow this. So what we are going to do is, this was a whole muscle, right? This was a fascicle, right? This is a single muscle fiber, got it? Which they've blown up right here for you, okay? So now what we're going to do is we're going to look at that even more closely. So this is a whole muscle fiber, okay? Now, this is a cell, but we can all agree that it looks weird compared to the 1610 cells that you looked at. The primary reason that it looks weird is because of its unique functionality, because it's contractile tissue. So all of these little Vienna sausage-looking things that are unique to this tissue, those are going to be related to its unique function of contraction. Good? But let's talk about the stuff that you have learned before to reorient you so that you can tell that this is a cell. So the whole thing is, of course, called a muscle fiber or muscle cell. Things that you have already known is that each individual muscle fiber is multinucleated, right? And notice one nucleus, two nuclei, three nuclei, right? Okay. And now can you tell why they tend to be peripherally located? Because is there room for them in the middle? No, it's busy in there, right? So what happens is they tend to get pushed up the side. So multinucleated, and they're peripheral, right? Okay. And striated. So let me see if I can show you where the striations actually occur based on, so you remember how striated it looked with the histology slide? Okay, so now let's see if we can figure out what that has to do with this. So look at, watch with me real quick. Do you see right here is a darker stripe going down there, and then a lighter stripe, and then there's going to be another darker one and another lighter one? Okay, so what we are going to see is the pattern that emerges in these things, these little sausage-looking things, is going to go dark, light, perpendicular to the long axis of the fiber. And so that's going to be the basis of striations. It's also going to be the basis of understanding how contraction occurs. So we'll get into that in more detail in just a second. Okay, so what else is true in there? An individual skeletal muscle fiber is microscopic in width, but can be up to a foot in length. The longest muscle in the body is, anybody know? Sartorius, yes. And it's not particularly long in me, but it's much longer in Orlando, right? He's much taller than I am, right? And then Shaquille O'Neal is impressive compared to both of us, right? So, and that longest muscle in the body will have long fibers up to a foot long in that one. Okay, so now let's start to dig into the fiber a little bit. First off, some bastard, and it wasn't me, renamed every internal cellular structure inside the muscle fiber. So instead of getting to call it the endoplasmic reticulum, you have to call it sarcoplasmic reticulum. And instead of calling it cytoplasm, they renamed it sarcoplasm. You don't even get to call it a cell membrane anymore because they hate us all. But I didn't do it, and I wouldn't teach them to you, except that I know that that's what they call them in other classes, so we can't avoid it. So I will do my best and just connect it to the thing that you've learned before. So first, let's start by saying, okay, the cell membrane around the whole thing is called the sarcolemma. It is just a cell membrane, phospholipid bilayer with embedded proteins. They're not showing the proteins, but they're there, okay? It is the sarcolemma. Now, let's see if we can figure something out. Conceptually, where is the sarcolemma with reference to the endomysium? Which one needs to be superficial and which one needs to be deep? What is superficial? Sarcolemma or endomysium? Yeah, right, because this is the boundary of the actual cell. Right? The endomysium has to be superficial to that, right? So wrapping, not shown on this figure. I just want you to understand that this actually has something to do with what we just covered, okay? So the sarcolemma is deep. This is the actual cell membrane. And then inside the sarcolemma is a bunch of stuff. Some stuff that's familiar, some stuff that's unfamiliar. Multiple nuclei, we got that, check. And then multiple mitochondria. This tissue, compared to other tissues, tends to have quite a few mitochondria. And that should make sense because what is mitochondria for? Energy, exactly. So we're going to have really energetically expensive tissue. And we're going to have the mitochondria that's necessary to do that. So basically what we need is ATP, right? And some other byproducts. And the place that this occurs is, of course, primarily in the mitochondria. We're going to have some other things that are related to that. Two things that you can't see in this image but are structurally or functionally important. Not structurally important. They are in what we call the sarcoplasm. And that's just the cytoplasm or the intracellular fluid of a muscle cell, sarcoplasm. Okay? All of this stuff is in the sarcoplasm. Two things that you can't see in the sarcoplasm but help you to really understand what this tissue is all about is a protein called myoglobin. Myoglobin is similar to hemoglobin. Hemoglobin binds to and carries around what in the bloodstream? Oxygen. Myoglobin binds to and stores small quantities of oxygen inside the sarcoplasm. So what we are going to see here is I've got some oxygen. I'm going to have something else so I can make some ATP. I'm always going to store this. The other thing that's stored in there but not shown on the figure, still functionally important, is glycogen. Anybody know what glycogen is? Glycogen is how animals store sugar, specifically glucose, yes. So now what we have is stored sugar, stored oxygen, and mitochondria. So what I am set up for is always, always being able to do some emergency cellular respiration, yes? So this is my emergency response tissue. A bus starts coming at me, even if I haven't eaten in 72 hours, I can still get out of the way of the damn bus because the last thing I did after I ate was to put that stuff back, yes? To make sure it's always set up, not for minutes of activity, but for seconds of activity, right? To make sure that you can always use this tissue in a pinch. Does that make sense to you guys? Okay, and now, of course, we have to address, one more little thing about myoglobin that's kind of cool. Myoglobin, when oxygen binds to myoglobin, it also makes it red. Just like when oxygen binds to hemoglobin, it makes blood bright red. So within the sarcoplasm is myoglobin. When it's fully oxygenated, the tissue is red as well. So now we have two reasons that your steak is red. The capillaries around each cell and the myoglobin within each cell, right? And I want you to think about that for just a second. If instead of cooking your steak, you accidentally get distracted and leave it out on the counter for hours and hours, maybe overnight, and come back tomorrow, what color will it be?
Brown.
Yeah, it's because the oxygen will dissipate off of the myoglobin and the hemoglobin. Does that make sense? Or you can do it faster and just cook it, right? And then the oxygen will dissipate off the myoglobin and the hemoglobin. Good? Okay, so now let's address these little buggers. These little Vienna sausage looking things. Go ahead, Mazzy.
So they like when your like CNS goes into like fight or flight, this is what's happening, right? Is these muscle fibers are
That is a very small part of what's happening, but yes.
okay.
Yes. There's a million things that are happening, but yes, that happens for sure. Okay, so what are these things, and what are they going to have to do with anything? Well, these are what is really structurally different about this tissue compared to other tissues. They have these big cable-like bundles of proteins. Do you know what a coaxial cable is? It's a cable made of cables, or a wire made of wires, right? So that's what this is. So a whole one of these sausage-looking things is called a myofibril. Can you say that, please?
Myofibril.
Okay, so how is this related to contraction? Short version is this. The myofibrils attach to the cytoskeleton at the end of the cells, and then when you stimulate it, they will shorten, and so they can shorten the whole cell. Does that make sense? Okay, so let's talk about how this is related to your experience with your own muscles. So I will have more contractile strength in a cell if I have more myofibrils and more of the proteins that make up the myofibrils. So let's talk about working out. So there's a certain type of working out you can do that actually will build muscle mass, right? Okay, so I'm going to talk about what the hell that has to do with this picture. So these are your contractile proteins, and they're made of little ones that are little bitty contractile proteins. And so I have X amount. I don't want to count them. But what I can do is, depending on what kind of training you do, you can increase the contractile strength of your muscle, yes? If you do the kind of training that you already know to do, right? Which is high weight, low rep, right? what you need to do is somewhere near the maximum of your capacity, meaning that Anthony's probably not going to lift 20 pounds with his biceps, right? It's not going to do that, right? Okay? But, you know, like a 10-year-old could lift 20 pounds, and that would be at the end of their capacity. So what you're going to do is you're going to try to get above 65% of your maximum capacity, and then you're going to do a few reps. If you can do 20, you're not high enough with the weight. And what is happening when you do that is you are communicating to your muscles. And what you are saying is, I need more contractile strength of you in the future. And here's what's going to happen. If you rest that muscle and feed it what? It will make some more of these for you. What do you need to feed it? Proteins. Because those are proteins. And you need proteins in your diet to make proteins. Yes? Does that make sense? Now, why will it work better in him than it does in me? Because testosterone facilitates protein synthesis in skeletal muscle better than does estrogen. Yes? Does that make sense? Okay. So what you do when you work out is you communicate with your muscles, and then if you rest them and feed them, they will do protein synthesis for you. So the real difference between Anthony before he started working out and Anthony after he started working out is not that you get more cells, because you don't. You actually are born without about all of the skeletal muscle fibers you're ever going to have. But what you can do is inside each fiber, you can make more contractile proteins. Yes? Generating more contractile strength. Does that make sense? Okay. It's called muscle hypertrophy. Good? Okay. So these little guys right here are called your myofibrils. And you can definitely make more of those. Was there a question? No? Okay. So let's break down the myofibril and see what it's made of. Okay. So going into the myofibril, follow with me because I'm going to have to go 3D to 2D and change pictures a bunch of times to make this make sense. So see if you can see what I'm talking about. So look at this myofibril right here. Do you see this jagged line? Yeah? Do you see that there's another one hidden under there? That jagged line hidden under there? Okay. What that is going to be is it's this repeating pattern of proteins that you will see in a myofibril. Think if the myofibril is a train, then this is individual boxcars. So let me blow that up so that you can see it, and then we're going to go a little simpler. Do you see the boxcars on the train up here? Yes? Do you see them? 1, 2, 3, 4. Yes? Those are all in a single myofibril. Everybody good with me? Okay, so now what I'm going to do is I'm going to take that 3D and I'm going to simplify it and look at it in 2D. Jagged line, this is one boxcar. Do you see? Okay, but we're going to simplify that and draw it on the board so that you guys can actually see what it's made of. Everybody okay with that? Go ahead.
So the light and the dark is what you're talking about in the striations?
Well, we haven't gotten to striations yet, but we're about to. Okay. So within each boxcar, there will be a dark part in the middle and a light part at the edge. Yes, and those will be the striations. Yes. Okay, so we're going to build this together, only a little simpler. Okay, we're going to concentrate on the anatomical structure instead of the physiological structure. Okay, ready? Okay, so what we're doing is we're building a single boxcar, and they're going to line up one after the other to make a myofibril. Okay, only this is a 2D picture. So what we are going to do is see the jagged lines at the edge. Yeah. From one jagged line to the other jagged line is one boxcar, which is called a sarcomere. Everybody okay with that? The reason we are doing this is because this will help you understand the physiology of muscle contraction, so we have to get some basic anatomy down first, okay? So jagged line to jagged line is a sarcomere. Draw that one better. Good? So here to here is a sarcomere. And now we're going to add some detail to it. Not as much as they have, because some of that isn't super important until you get to phys. So first thing that we are going to do you see these red string of beads looking proteins right here? Okay, notice that they're attached to the jagged line. Oh, by the way, I need to identify the jagged line. The jagged line is called a Z line or Z disc. The phys book uses one and the anatomy book uses the other. If it's got Z in it, that's what it's talking about. Okay, attached to the Z lines, extending toward the middle, but not generally touching, are these red proteins that I'm drawing in red. And those are made of a specific protein called actin. And it is the thinner of the two that we're going to learn. Okay, know. So the protein is called actin. It's the thinner of the two. Break down myofilament to me. Myo meaning muscle, and filament meaning long skinny thing. Okay, so it's a long skinny protein that makes up muscle. Yes? Okay. And then floating in the middle, actually not floating, but it looks like it's floating in the middle, is a much thicker protein. Do you see it? Right here? See that one? Okay. They've got three. I'm only going to draw two. This is the myosin. Myosin. It's the thicker of the two. And it's also a myofilament. If you can give me just a second, I'll be able to explain how this causes contraction and also striations. Okay, two other little things to add, and then we won't add the rest. Okay, do you see that myosin has these little projections sticking off of it on the picture? Yeah? Okay. That is called the myosin head. And then not shown on here, but I'm going to draw it just so that you can have a visual for it, okay? There's these little binding sites. They don't look like handles, but just we'll go with the metaphor so that we can learn. Okay, this is an active site. Okay. So let me give you a preview of the complexity of muscle contraction, just a simple version. So how do you cause a muscle to contract? Well, it starts at this level because in order to make a muscle contract, what you need to do is stimulate this thing. Don't worry about how the stimulation occurs at this particular moment. And then what's going to happen when you stimulate it is the myosin heads are going to grab the actin, active sites, and they are going to pull, right? And so when that occurs, what's going to happen to the distance between the Z lines? It's going to shorten, okay? So let's just identify the whole striping pattern, then we'll figure out what the stripes do when you contract the thing, okay? So as far as the striping pattern goes, so watch me for just a second here and you can see how this occurs. So if I use a protein staining technique, wherever there is more or denser proteins, then it's going to stain darker. And wherever there are fewer or less dense proteins, it's going to stain lighter. So see if this makes sense to you. Do you see that this area right here is going to be the darkest area of the sarcomere? Yes? Why? Because this thick myosin protein is in the middle. Yes? That makes sense? So this right here is called the A band. It's the darkest band. Think D-A-R-K, second letter in dark. Okay? And from the edge of the A band until the edge of the next one, isn't it going to be lighter? Can you see that it would be lighter? Because there's only this thin protein comparatively. Okay? So that is going to be from here to whenever it hits the edge of the next one is going to be the I band. And last thing, and then I'll show you how this works. Yeah.
Where's that light line
From the edge of the A band
Okay.
until the edge of the next A band, which you can't see. So it's just from here. Wherever there is no myosin, yeah, is going to be the I band. Okay? Then one last little thing. Does it make sense to you that in the middle of the A band, it's going to be dark but slightly lighter right here? Right? Because you don't have overlapping of these two proteins. So right in the middle, right there, is going to be what we call the H zone. And please note, it's already labeled here too. So the H zone right here, it's going to be not dark, but not as dark as the rest. Okay. A band, I band. That is going to correspond to striations. So let's look. So this is using a different protein staining technique, but let's just see if you can find the actual things that we're looking at. Here's the Z lines right here, the edge, right? Here's the A band, the darkest area. Notice in the middle, there's an area that's not quite as dark as the rest of it. That's the H zone. Good. And then as Anthony just asked, the I band is from the edge of one A band into the edge of the other one. Okay, so how does this correspond to this? A band, do you see it? Then I band, right? Then A band, then I band, all the way as you go through, okay? Since these line up so perfectly, they end up making the entire tissue striped, okay? That's the nature of the striation. So let's see if you guys understand it by contracting it and asking you what happens to the bands and the zones. You ready? Okay. So I want you to sit here and stare at this one, okay, or the picture that you drew, and tell me when this sarcomere contracts, okay, which of the bands and zones are going to change length? Stare at it until you come up with your hypothesis. Which of the bands and zones are going to change length when this sarcomere contracts? Remember that how the sarcomere contracts is the myosin heads reach up and grab the actin sites and pull. So what's going to change?
The A band is going to get short.
A band is going to stay the same. A band is in the middle, just sitting there pulling stuff into it. H zone will shrink almost completely if you contract maximally. What else? And the I bands are going to shrink. Does that make sense? Yeah? Can you see that? Okay, so let's look. Here's a figure that shows you just that. So let's start with the top. This is a relaxed muscle. Try if you can to pay attention to the H zones and the I bands on each one, okay? So you see the H zone in the middle, H zone in the middle, 2 H zones over here, right? 2 I bands, right? Then partially contract it, right? What happened to the length of the H zone? It shrunk, right? What happened to the length of the I band? It shrunk, but just a little in this one, right? And now let's get maximally contracted. See if you can find any H zone at all. Okay, maximally contracted. Do you see any H zone at all? No, it's completely contracted, right? Even though this one looks a little lighter, it's just because it was stained with too much light. Don't worry about that. So basically, it's all really uniform. Go ahead, Bridget.
So I-bands aren't actually getting smaller, just being brought closer together.
Exactly. So what's happening is the filaments, this filament and this filament, these two are sliding past one another. So they call, it's a great intro question, it's almost as if I fed it to you. So what they call this is they call this the sliding filament theory of muscle contraction. Yes? Because what's happening is in order to contract the muscle, does actin actually change length? No. Does myosin change length? No. What they do is they slide past one another, right? And now we can actually confirm this because we can stain actin and myosin 2 separate colors and watch them slide past one another. But interestingly, before they were able to do this, what they actually looked at is on a microscope, you look at it under a microscope, if you have a living piece of muscle under a microscope and you stimulate it, it'll go from being striped to being almost completely dark, to being striped to being almost completely dark. And so they hypothesized, even before they could stain them independent colors, that the filaments were just sliding past one another. And this is actually how, when I took physiology, when I was in college as an undergraduate, we still did animal dissections in physiology. And I did a frog gastrocnemius. And I understand your objections, but I will tell you that I know this really well because of that learning experience. Because what we did in my physiology class was we pith the frog, which is anesthetize the frog, pull out the gastrocnemius muscle, stick it on a special slide and attach the points, and then stimulate it with an electrode, right? And watch the thing go from being striped to being completely dark, to being striped to being completely dark, striped, completely dark. And yes, I understand that there are good reasons not to do that anymore. However, I know this better than you probably ever will. Because of that experience, right? Because of the experience of actually having done that. I mean, some of you guys will know it super well. So anyway, I can tell you that it was actually kind of shocking and a momentous occasion for me to like put that together with this. And I never really forgot it. Okay, so, no. Well, we used both legs. So the frog was anesthetized as we did. It wasn't just one person. So we used all the muscles that we could. And then the frog was euthanized. So yes, I understand your objections. I'm a total animal nut
Is there
too.
like an animation where you can see that?
I've never looked. Will you send me an email? I'll look for one.
Yeah.
Yeah. Mm-hmm. Okay. So The basis of skeletal muscle striations is therefore sarcomere anatomy. So why is it striped? It's striped because of the sarcomere is striped. But the sarcomere is also the basis of contraction, isn't it? Right? So it's all related to one another. So the basis of skeletal muscle contraction we call the sliding filament theory, but just to go through it one more time. First of all, none of you guys are those people who argue that theory doesn't mean that we don't know if it's true, right? You're not those people, are you? Because everyone laughs at you online if you say that theory means that we don't know if it's true. Theory in the life sciences means there is a preponderance of evidence to support, and there's no evidence that we found yet that will overturn it, right? But we don't have laws in the life sciences. Those are in the physical sciences. So sliding filament theory, cell theory, evolutionary theory, preponderance of evidence. So the sliding filament theory of muscle contraction says that if you stimulate a muscle with it's a specific type of neuron, if you stimulate it with a specific type of neuron, the actin and the myosin filaments will slide past one another and the Z lines will come closer together. The H zone and the I band change width, but the A band stays the same, pulls everything into it, right? The filaments don't shorten, but the sarcomere does. So the sliding filament theory of muscle contraction. Does that make sense? Yeah? So this is the whole basis of the phys. Now, when you get into phys, you will figure out that there's about 3 more proteins that you have to learn, and there's a whole in-between part between what happens between the stimulation and the contraction. And the good part is that it's interesting as hell. Physiology is absolutely fascinating, but it's not very easy, right? It's a different kind of hard than anatomy. Who in here is in my phys class, I think? Oh, there's 3 people in the other class. Oh, she's not here. There's 3 people in the other class that are in phys. Phys is a different kind of hard than anatomy. It's not very memorizing. Go ahead.
When your muscles contract, is that when like lactic acid gets released?
If you do not provide it with adequate levels of oxygen, then it will do anaerobic respiration and release lactic acid and then run out of energy really quickly. Yes. Good. How'd you learn that, Bridget? Where'd you learn that? Oh, good. In when? In high school? Biology? Oh, good. Oh, good. I didn't know they taught it. I was like, I remember 0 from high school biology. I remember that there was a really cute boy 3 seats down from me, and that my teacher had super bad breath. I remember that too. Okay. So let's do a macro to micro organization of skeletal muscle, all the way from a whole muscle down to those filaments, right? And then we'll do it all the way back up, and then you guys will be bored with it, which means you probably are ready to actually say that you know it. Okay, so we're going to start all the way from a whole muscle. I'm going to ask you guys questions. You ready? So a whole muscle, the whole thing, is wrapped in connective tissue called what? Epimysium, yes? Okay. And is composed of multiple bundles of muscle fibers. A bundle of muscle fibers is called a what? Fascicle. Each fascicle is wrapped in connective tissue called what? Perimysium. Good. And is composed of multiple muscle cells or muscle fibers. Good. Each muscle fiber is wrapped in connective tissue called endomysium. Good. And the boundary of an individual muscle fiber is the cell membrane or sarcolemma. Good. Inside the sarcolemma is a bunch of other stuff, but we're going to concentrate on the contractile proteins, okay? Inside the sarcolemma is a cable-like bundle of contractile proteins, a whole bunch of them. Each cable-like bundle of contractile proteins is called a what? Myofibril. The whole thing is a myofibril, and it's composed primarily of 2 myofilaments. The thinner myofilament is called a what? Actin, and the thicker myofilament is called myosin. The repeating pattern of actin and myosin from one Z line to the other is called a what? Sarcomere. Good? Sarcomere. Okay, you ready to go from the bottom to the top now? Yes, you are. Okay, so how do I make muscles contract? Primarily with 2 really skinny contractile proteins, right? The thicker of the 2 is called what? Myosin. The thinner of the 2 is called actin. The repeating pattern of actin and myosin that gives both striations and allows us to understand contraction. What's that repeating pattern? The boxcar on the train. Sarcomere. Okay, add up one sarcomere after another. Boxcars, the whole train that it makes is called a what? Myofibril. Okay, myofibril. A whole bunch of myofibrils plus all the other organelles is wrapped in a cell membrane called the sarcolemma. Superficial to the sarcolemma, you run into your first layer of connective tissue in muscle called what? Endomysium. Good? Okay. Take a whole bunch of muscle fibers, wrap them into a bundle. The whole bundle is called a what? Fascicle. And it's wrapped in connective tissue called what? Perimysium. Take a whole bunch of fascicles, make a whole muscle out of it, and wrap it in connective tissue called what? Epimysium. Good. That make sense? Okay. That's the relationship of all of it. What?
Again.
Again?
Drill it.
No, you do it.
Not ready.
I'm not ready.
I wouldn't complain if you repeated that.
Okay. All right.
Because I have a feeling that's going to be on the test.
It's like really important for anatomy and for phys. So, okay. So, which way am I going to do it? Which way is harder?
The hard. I think the
Small to big. Okay. I have no idea how I said it before, and I never say it the same way. So just,
process can how muscles.
I don't know. Stuff with things, structure, micro to macro structure in muscles. And every time people goes, will you repeat that? I'm like, I have no idea. That was nanoseconds ago. I don't know what I just said. So at least you know I'm not just reading off the PowerPoints, because I don't read off anything, because I don't know what the hell I'm going to say from one moment to the next. Okay, so we're going to build a whole muscle. And we're going to start all the way down. You don't have to write it, because it'll be on the recording, you know. Okay? You could just chill and think. So we can start all the way down at the submicroscopic level. Yes? I mean, you can write if you want to. I'm not your boss. You do what you do, you, man. Okay. So all the way down at the submicroscopic level, we have two contractile proteins. Yes? These two contractile proteins are the thin one that is attached to the Z lines. What's that called? And the thick one, which isn't attached to the Z lines, myosin. The repeating pattern, the stripes that actin and myosin make from Z lines to Z lines is called what? Sarcomere. Add up one sarcomere after another until you get this cable-like bundle of contractile proteins. That cable-like bundle of contractile proteins is called a what? Myofibril. Take the myofibril, put a whole bunch of those, some other organelles in there. You make a whole muscle cell or muscle fiber. Yes? The boundary of the muscle fiber is the cell membrane or sarcolemma. Superficial to the sarcolemma, you run into your first layer of connective tissue. What's that called? Endomysium. Now, take a whole bunch of muscle fibers together, and they make up a group of muscle fibers or a bundle called a what? Fascicle. The fascicle is wrapped in connective tissue called perimysium. All the fascicles together make up a whole muscle, and that's wrapped in connective tissue called what? Epimysium. Good. Yeah. See, it's not that hard. You got it. Yes. Okay. So, that's not what I needed. Where are you? Okay. So now let's just compare and contrast to cardiac muscle and smooth muscle tissue. The hard part's over. So cardiac muscle tissue. You know what cardiac muscle tissue looks like on a slide. Does it surprise you at all that cardiac muscle tissue also has sarcomeres, actin, and myosin, because it's also striated, right? So the good part is that all of the trouble you go to understand skeletal muscle anatomy helps you for cardiac muscle anatomy, and also skeletal muscle anatomy and cardiac muscle physiology, right? It helps you for all of it. Okay, so it has some things in common with skeletal muscle. First off, it's striated, right? It has the same arrangement of actin and myosin. It also has lots of mitochondria. However, it is always involuntary. You cannot control cardiac muscle voluntarily, meaning that if you get super nervous about something and it's not an opportune moment for your heart rate to be 120 beats per minute, can you just go, hey, stop? No, right? All right, wouldn't it be great if we could just like control our actual tissues and make myself ever chill at all? But no, I've given up. It's involuntary, so you can't control it voluntarily at all. Differences anatomically, shorter fibers, yeah? One nucleus instead of multiple nuclei per fiber, and tend to branch, right? So they branch and talk to one another. And then we've got these intercalated discs, right? And the intercalated discs are the site at which you have two junctions. The one that makes it strong, which is what? Desmosomes. And the one that makes it communicate, which is? Good. Desmosomes and gap junctions. So shorter fibers. Okay, those are intercalated discs. So that's the primary differences. Okay, and then the last thing is just smooth muscle tissue. And smooth muscle tissue looks like it would have a completely different physiology and contractile mechanism, but it actually doesn't. It's just that the actin and the myosin don't line up like one another. So let's look at this picture for just a second. So what you have, and I won't make you label this one, I just want you to see the similarities between what you've already learned in this. What you have is I still have myosin, that thick filament, and I still have actin. But since all the myosin doesn't line up, then I don't have an A band, right? But is it still going to have a really similar contraction mechanism? It is, right? It's still going to, myosin heads reach up and grab the actin active sites and pull. Only, of course, that since they're going a bunch of different directions, and it doesn't just pull in length, it also pulls in width. So the cell kind of squishes together when you do that. Does that make sense? Oh, balls. Just kidding. You don't have to be sorry. It's not that big a deal. Okay, so it's got some similarities. It's got some differences. It's found any place that you have a hollow organ or a hollow tube, except the heart, right, that you need to change the lumen size or move things through it. So organs and tubes, bladder, blood vessels, uterus, all those locations. Non-striated, and again, it's not striated, not because it doesn't have actin and myosin, but because the actin and the myosin do not line up with one another, okay? It's still involuntary, right, like cardiac muscle. And then the functional anatomy of smooth muscle in hollow tubular organs is generally like this. So stick with me for just a second. If I have a hollow tube, imagine that this is the esophagus. What I usually have is two alternating smooth muscle layers. I've got a circular layer that's usually close to the lumen. When I contract that one, it narrows the lumen, right? And then I've got a longitudinal layer, and the longitudinal layer is usually more superficial. When I contract that one, it pulls things through. So how do I make things move through tubes? I alternate squish it tight, pull it down. Squish it tight, pull it down, right? That's peristalsis. That's what makes things move through your intestine, right? So those two act kind of alternating to one another, and then they produce these wave-like movements. It's kind of a gross one, but you need something gross today, don't you? Want to see peristalsis move things through the intestine?
Yeah.
Ooh, yeah, look at that. Sexy. If you've ever seen a snake eat, you can kind of see it getting skinnier and squishing back. That's intestines. Are you sure you want to go into human health? Humans are gross. Ooh, isn't that one awesome? I love that one.
That's like
Okay.
white, but like.
It's fat.
Oh, it is fat.
Mhm.
Okay.
Yeah, it's fat. Okay, so we'll stop there for today, and we will do skin next time.